lecture we are having an observation towards the enzymes, also of the denaturation of proteins. In today's lectures, the biomolecules. We are going to study about the nucleic acids. This one is very interesting point of the biochemistry. Nucleic acids, these are present generally in the nucleus, also in the cytoplasm. Nucleus, those are which present in nucleus and in the form of acid, or those present in nucleus, and these are, when we observe, these are negatively charged species. These are have the negatively charged phosphate group in their molecules. So generally these are considered as the nucleic acids. When we observe towards the nucleic acids are the remarkable properties of living cells. Their ability to replicate through thousands of generations. Replication, I am taking here the word replications of thousands of generations. This becomes possible because certain types of information which is passed through from one generation to another generation. Such information is called genetic information. The information which is passed through one generation to the next generation, it is genetic information, and it is transferred to new cells which are accomplished by nucleic acids. When we observe toward the synthesis of proteins is a fundamental structural material of body. This process in which genetic information is transferred. When we observe the genetic information which is governed by the DNA present in the chromosomes of the cells of the nucleus. Each chromosome has a different type of RNA. Each chromosome has a different type of RNA. When we observe the process of preparation of proteins, DNA, which is the base of the genetic information, RNA, which transfers that information, and we are having the proteins, which are the fundamental material for the process of life, process of building of the body, or the formation of the genetic information, which are come one generation to next generation. The species, observe the human being, the dogs, the cats, the wild animals. Also we are having the species like the trees, mango trees, banana trees. These are have the specific structures. These structures are present in between their cells in the form of DNA, in the nucleus. When we observe the genetic informations which are required there, nowadays we are having the knowledge about the DNA test. The one person's DNA is come from his parents, 50% from its parent, mother, 50% from its parents. So we are having the confirmation of the species. The person, it is the son of this and this. This is possible due to the information or the knowledge about the DNA. When you observe the DNA as well as RNA, DNA it is the RNA firstly, RNA. It is ribonucleic acid, and DNA is deoxyribonucleic acid. These both DNA and RNA both are found mainly in the fluid of the living cells, which is considered as RNA, which is present in the cytoplasm, DNA present in the nucleus of the cell generally. But these both are present in the cells. When we have to study about the RNA and DNA, the word ribo it is the word for the ribose sugar, and drawing a structure of the ribose sugar in the form of pheromones. Here, the ribose sugar this one is beta D ribose structure, and we will observe the basic ribose sugar present in DNA molecule. It has the absence of the hydrogen group in the main sugar unit. This have the absence. This one is beta 2 deoxy, beta D 2 deoxy ribose. This one is the basic of the RNA, and this is the basic of the DNA molecule. When you observe towards the DNA as well as RNA, these have DNA and RNA molecules have repeating monomeric units, and these are the nucleic acids have the unbranched polymers of repeating monomers called nucleotides. First one, we have to know about the nucleosides. Nucleoside, it is generally the base and sugar unit. Nucleoside, when we observe nucleoside, it is the here we are having a presence of base. This one is the base and sugar unit, which is present in nucleoside. And when we observe the nucleoside, base, sugar, and a phosphate group is present. There is presence of hydrogen, but I am removing it. Hydrogen is removed here. We have the presence of negative charge ions. This is a structure of nucleotide. This one is structure of nucleoside. This one is structure of nucleotide. It means nucleosides are base or we can say sugar plus base unit nucleoside, and sugar plus base plus phosphate group is known as a nucleoside. Nucleoside is the unit of sugar and base, and nucleotide is a unit of base, sugar and phosphate group. Simple. Phosphate group, when we react H3PO4, it have a removal of water molecule at this carbon. Here, this group is removed here, H2 in the form of H2O, and we are having the presence of H2PO3. H2PO3. Here hydrogen is removed to have the formation of corresponding nucleotide. This bases, observe in the presence of nucleotides as well as the nucleosides which have the presence of base. When we study about the DNA or RNA or the nucleic acid, we are having 5 bases. We are having 5 bases that are adenine, cytosine, guanine, thymine, and uracil. These are the 5 bases. When we observe towards the presence of nucleotide units, which are thousands of units present in RNA, and several millions of units of nucleotides are present in the DNA. So here, firstly we have to draw the structure of these 5 bases. So we are drawing here bases of adenine, cytosine, guanine, thymine, uracils, which are present in the DNA as well as RNA in the nucleotide chains. First of all, so these 5 bases, adenine, cytosine, guanine, thymine, uracil, these 5 bases are present in the molecules of DNA as well as molecules of, or we can say the RNA. Only we are having a difference about the presence of thymine or uracil. DNA has the presence of adenine, cytosine, guanine, thymine, and RNA have the presence of adenine, cytosine, guanine, and uracil. It means thymine or uracil in these 2. When thymine is present in presents, it is present in DNA, and uracil is present in RNA. These are the structural representation of these bases. When we observe towards the monoaromatic ring, or you can say the only one ring is present in the cytosine, uracil, as well as in the thymine. These are have the parent compound or the structure. These are often from the pyrimidine structure. They have the structure of pyrimidine. Only one ring, which have the presence of this nitrogen base. When we observe towards the structure of uracil, adenine, these are structural resemblance with the purine. It means their parent compound is purine. When we observe towards the cytosine, uracil, and thymine, these are resembles the structure like pyrimidine. And when we observe the structures of adenine and guanine, This resembles the structure like purine. This one is structural representation of the bases. When we observe towards the formation of nucleotides molecule or formation of nucleoside molecules, we are having some different numberings. Some different numberings. Instead of the numbering C1, C2, C3, C4, we have the prime numbering. Prime 1, prime 2, prime 3. Like this we are having the numberings. These are due to presence of these bases in the RNA or in the DNA. So we have to know about the formation of nucleoside, also the formation of nucleotides, how these are formed, how the phosphate group is present, how they have the diester linkages in their molecules, how these are named. We have to observe towards this, the formation of nucleosides, dinucleosides also, where have the formation of nucleotide. So this one is the reaction of ribose sugar, reaction of beta D ribose sugar with the cytosine, which uses the water molecule to have the formation of a ribonucleoside. Here we are having attachment of the nitrogen with the first prime. This we have a simple confusion about the numbering of carbon atom. So the bases are numbered on the basis of the number letters 1, 2, 3, 4, 5. But when we observe, when we have the numbering to the ribose sugar or the sugar unit present in the nucleoside or the nucleotides, we use the prime 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. This prime is used for the numbering of the carbon atoms present in the sugar unit. When beta D ribose is reacted with the cytosine, it loses a water molecule to have the formation of a nucleoside, which is known as ribonucleoside. But when we observe the formation of a nucleotide, nucleotide is prepared from, when we observe towards the formation of these nucleotides molecules. Nucleotide molecules are the formation of nucleoside in the form of 2-deoxyribonucleoside. This one is the preparation of 2 or deoxyribonucleoside. Deoxy, and then name beta D2 deoxyribose is treated with corresponding adenine. Adenine loses a water molecule to have the formation of corresponding. What is formed here? Or it is we are having formation of 2-deoxyribonucleoside. This denotes the formation of nucleoside from the corresponding beta D2-deoxyribose. Also we are having the formation of nucleoside from beta D ribose. When we have numbering these compounds, how these are named, and when we how these are considered as the names, and this is the abbreviation into the further nomenclature or for writing the names of the corresponding nucleotides, also the nucleoside molecules. And this one is the nucleotide molecule, and we are having the presence of the phosphate group. The capital letter, first capital letter of this name, it stands for the name of the base, adenine. AMP, it stands for adenine monophosphate. Adenine monophosphate. This one is named as, listen carefully, dicytosine monophosphate. This one is named as dicytosine monophosphate. These are the namings used for the corresponding, or you can say the names of the compounds. You know, of the those they have. This one is the name of the DNA molecules. It is named as deoxycytosine monophosphate. Deoxycytosine monophosphate. The first capital letter, these are the name of corresponding base. MP stands for monophosphate, not for Madhya Pradesh. C stands for the name of base. MP stands for monophosphate. Deoxy, D stands for deoxy. We are having the absence of hydrogen group at the 2' carbon. So it is named as deoxycytosine monophosphate. These are some examples of the DNA RNA's base, which is known as the nucleosides or nucleotides. So, this one is the general information about the basic units of the corresponding nucleic acid molecules. The nucleic acid molecules have the presence of nucleosides, nucleotides. These have the attachment with each other by the phosphate linkage. How these are linked together? We have to know about the linkings. You know, observe the linking towards the present in the RNA, DNA molecules. The Watson and Crick in the 1953 Watson and Crick model of the alpha helix, or we can say it as double helix structure. Double helix structure of the DNA molecules takes place there. Here, DNA, RNA, which are have the presence of nucleotides, units, nucleotide which is known as the base, sugar, and the phosphate group, and nucleoside which have only the presence of base and sugar. Base and sugar, nucleoside. Nucleotides have the presence of base, sugar, and phosphate group. These are examples of nucleotides. These are made by using the abbreviations, where the first capital letter stands for the name of the corresponding base. Here adenine, here cytosine. These are the name of the bases. First capital letter, and MP stands for monophosphate. And when we observe the actual small letter B, which is used for the indication of corresponding deoxy. This one is has the presence of hydrogen group, so it is ribonucleic acids or ribose structure. The sugar it is ribose here. The sugar it is 2-deoxyribose. Now we have to know about the structure of the DNA, also about the structure of RNA molecules. Simple structures are there. Two chains in DNA, we are having two chains of the corresponding bases. It is very useful.